Thank you. So that is really the take-home message from this lecture, Motif Matters. I should have a little flag. And I want to thank since I see you, Polly, thank you for introducing high-throughput biochemistry as a very important thing to do to complement the genetic screening approaches. Yes, and I am actually delighted to be here. I love Barcelona, and this community is new to me, so I'm happy to meet all of you. Before I go into my talk, I want to mention my lab. It's really an amazing team. They are very hardworking and very brave. They always propose activities that sometimes they propose activities that I don't like to do, like climbing on the roof of Uppsala Castle. But most of the time we do things that I'm also excited about, such as exploring the human interactome. So if you think about the human interactome, it is currently being charted by a lot of different approaches. So we have, for example, yeast to hybrid approaches providing information on direct interactions. We have APMs providing information on complexes, and proximity-based approaches providing information on if proteins are close enough in space. So a massive amount of data is out there in the databases, such as intact, biogrid, string, and so on. But still, I would claim that a lot of data is still missing. And of course, as a motif propagist here, I would say that the motif-based interactions are among the most uncharted. So what are these motif-based interactions, or short linear motif-based interactions? Well, they are essentially a couple of key residues that dock into a binding motif, a binding pocket. So we can see here a p-type motif, which is being bound by the UEV domain of TSG101. And as you can notice here, it is the same human binding pocket here is binding both to human and the viral ligand. Now, these motifs are typically found in the intrinsically disordered or unstructured regions of the proteome, which are still a bit difficult to study. Now, these interactions, we would say from our side, is that the motif-based interactions are crucial for cell function. So they are, for example, important for bringing together complexes at the plasma membrane, for signaling, for cell cycle regulation, and so on. Still, as I said, we do miss a lot of information. Why is that? Well, the main reason is probably that they are occurring with fairly low affinity, and they are transient, meaning that you typically have a rapid off kinetics so that they are easily lost when you are conducting a lot of washing steps in, for example, your pull-down experiments before a mass spectrometer, analysis by a mass spec. If we look at the databases today, the gold standard database is the eukaryotic linear motif database, which contains about 4,300 interactions. Now, this is a wonderful database, which is a gold standard database for the field, but it does have a limitation in that it's not very comprehensive. So what we have been doing lately, together with Norman Davey, is to build a new database, which we called the motif map of the proteome. Now, when I say we, I mean that Norman built the database and that my lab contributed with curating a lot of information. I think I am a little bit crazy, so I think I went through about 1,000 papers myself. Things that you can do when you're bored, right? But still, what we are doing now is trying to answer a couple of questions. And one of the open questions is, how many motifs do we have in the human proteome? We don't know. It has been estimated that there are about 100,000 to 1 million motifs in the human proteins. We are also wondering how many motif binding domains do we have, and again, we actually don't know. There might be 10,000 of them, maybe. But we think that within the next 10 years or so, we will figure this out. What is important for this meeting is, of course, how are these interactions regulated and deregulated? And that is also a very important question. The workhorse method that we are using here is the proteomic peptide phage display, which is really a dedicated method for large-scale analysis of motif-based interactions. And together with Norman Davey, we have been developing this method over the last 10 years. And as it works is that my lab is an experimental lab, and Norman's lab is mainly a computational lab. So together we kind of make a complete lab, but in different countries. The phage, for those of you that are not familiar with it, is a virus that infects and replicates in bacteria. And by linking the peptides that we want to screen for interactions with the major coat protein P8, we can get this multivalent display on the phage coat. Now, the multivalency is important because the interactions that we are trying to capture are fairly weak. So if we have tried, I should say, we have tried to put the peptides on the P3 protein out here to get a monovalent display. And then we can capture interactions for some proteins, but for most not. You may wonder, phage display, that is a fairly old technique, right? Not very exciting. It was first developed in 1985. I got a Nobel Prize in 2017. But our twist here is really in the library design, which has kind of changed the scene. So what we do is, instead of taking randomized peptides, and I should say that we also have random peptide phage libraries, but instead of taking them, mostly what we do is to design highly specific phage libraries, where we take, for example, peptides from the human proteome or from viral proteomes. And then we fuse them together with the P8 protein. What we also do a lot of the time is to produce a massive amount of bait proteins. Well, it's massive for our hands. And I agree with this previous speaker earlier today saying that the constructs here are very important. So we do spend some time optimizing constructs. When we have our bait proteins, we can do our screens here in 96-well formats. We come with our phage libraries, we let them bind, and then we wash away what is not binding. We elute, amplify, and sequence. So the readout is next-generation sequencing, which is fairly cheap for interactomics approaches. And then what we have built is really a complete data analysis pipeline, where we take the NGS reads and then map them back to the proteomes and assign confidence levels so that we eventually can start to map protein-protein interaction networks. When you generate a lot of data, you also need to have somewhere to store it. So we have also generated the proteomic peptide phage display database, where we deposit all our data. And as we publish studies, we also release all our data here. I should say we do deposit all interaction data, the highest quality interaction data in databases such as UniProt and BioGrid. But since we think it may be useful for the field to have access to all data, we also put everything in here. So here you can, for example, see that in this selection against the protein G3BP1, we pulled out this peptide in 3 different selections, and so on. So it's quite a rich database. Now, how did this start? It all started actually back in 2012 or something, when I was in Belgium, actually, doing my second postdoc, and someone said, well, I was hoping to start my own group, but I didn't get the funding, so I said, okay, I have 1 year left in science, so let's do something fun. So I went to Canada, and I made a last postdoc with Dev Sidhu and Philip Kim, because I was very inspired by this idea of being able to take all the C-terminal parts of the human proteome and put them on a phage library to fish out binders for PTZ domains. So this worked pretty well. So we later then, when I started my own lab, we developed the first generation human disorderome, where we looked at all the disordered regions of the human proteome. And we showed that we can also develop a more general tool. So that was the first generation. More recently, what we did was to optimize this resource so that we now have a phage library where we have tiled all the disordered regions of the human proteome with 16 amino acid peptides with an overlap of 12 amino acids, which we then can use in high-throughput studies. We have benchmarked this approach using a target set of domains. So we picked 35 domains from that were listed in the ELM database as being known peptide binders. And then we subjected them to our property screening. And we could then show that our recall is about 20% of all expected motifs. And that might sound low, but it's actually high. It's about twice the recall of motif-based interactions as you would get using APMS or yeast-2 hybrid. What we have also shown is that we can capture a wide range of affinities from low nanomolar, as shown here for the KEAP1 Kelch domain, up to millimolar, as shown here, for the KPNB1. So binding is one thing, but function is maybe, well, it's also important. Well, they may or may not come together. So the question was, of course, if these peptides that we bind are also functional in a cell. So to answer this question, we focused on the nuclear localization signals that binds to KPNA 1, 2, 3, 4, 5, 6, 7, the important family. And using then KPNA4 as a bait, what we did was that we fished out quite many different peptides that we could show were binding either to the major or the minor group of KPNA4, and by fusing them to GFP, we could show that the peptides brought GFP into the nucleus. So we have kind of an efficient way for screening in high throughput for motif-based interactions. Yeah, I convinced you. Why does it matter to have all this information? Well, if you think about it, we are doing high-throughput interactomics analysis with the amino acid resolution. So we know the binding domains and we know the binding peptides. And by looking at the binding interfaces or the predicted binding interfaces, we can start to predict the effect of mutations. So here again, we are coming back to KEAP1. We can see that we found a bunch of peptides. Many of these peptides highlighted in blue here have phosphosites, or in boxes actually are the phosphosites, which could potentially regulate these interactions. But we can also see that we capture motif mutations that are, mutations, disease-associated mutations that are in the middle of the binding motifs. And based on that, we can start to predict that these mutations will likely then perturb the interactions. And of course, we tested this being an experimental lab. We measured the affinities. And what you can see here is that here we have our wild-type interaction between KEAP1 and NFE2L2. And here are the two mutant peptides. So this is a pretty powerful way to actually just, you fish out the binders and you check if the mutations are hitting on the binding sites. Another example is coming here. This is KPNA4, which binds, as I said, to the nuclear localization signals. And again here, we can see that we have one mutation, which is disease-associated in the protein NKX2.5, which is associated with atrial septal defects. Now, by testing this in the context of the full-length protein, we can see that if we introduce this mutation in the transcription factor, the transcription factor becomes, it loses its nuclear localization, which is likely explaining then the molecular disease. It's probably explained why this mutation causes a disease. So this is published, and I just want to tell you where we have been. And we have already released then more than 2,000 interactions, and we have shown that it's possible to predict the effect of disease mutations. Now, during the last couple of years, with a bit of time, we have worked on an upscaling of this, so going from 35 domains to somewhere between 600 and 800 proteins that we expressed and purified, and we generated a novel data set of about 28,000 interactions. And our novel data set will contain binders of more than 100 domain classes, and we hope to release it in August, September this year. I said this also last year, that I would release it in August, but 2024, but I think now we are actually ready to release it. Yeah, it's a bit of a challenge with our large data sets. So I'm not going to tell you about this because this is a variant effect meeting, so I thought I would talk about how motif-based interactions are actually affected by disease-related mutations. And I already told you that we can predict the effect of motif-breaking mutations by first finding the binding sites. But the more challenging part is actually to find mutations that make novel binding sites. It's quite difficult to predict. And a bit of background here in the field is that it has been known for quite some time that motif-breaking mutations exist and that they are linked to different diseases. And there are a couple of sporadic examples where we have disease-associated mutations that actually generate novel binding sites, which was beautifully shown in this study by the Selbach lab. So around, well, a couple of years ago, Johanna here started as a PhD student in my lab. She's a wonderful person if you ever meet her. She's currently doing her postdoc in Copenhagen, but the reason she joined my lab is that she really loved to work with proteins. She knocked on my door and said, I love to work with proteins, I need to work with you. So we decided that she should work on exploring how disease-associated mutations affect binding. So the key question here is that, okay, this here is the wild-type peptide. What happens if we introduce mutations? So what we did was simply to look into all the databases that we could think about and pick out all single nucleotide variants that had had non-synonymous effects, which overlapped with our human disordrome library design. So we designed a new novel library that we called the GenVar HD2 phage library, which then contains both the wild type and the disease-associated mutant versions of the peptides. So this library contains about 12,500 mutations and cover a wide range of diseases, so from cancer to neurological diseases. So we have a library. Then what we needed was a bait protein collection. So Johanna decided to purify 80 different proteins from many different proteins, protein domains from different proteins. And we thought that if we can use these baits in this selection, then we will ideally pull out a lot of disease-associated mutations that are breaking the interactions. And we were expecting to find a couple of mutations here that were enabling interactions. So when we got back the data, we were a bit surprised, actually, because we found more than 1,100 interactions. And of them, we found, as expected, a fair number of loss of binding mutations that appeared to perturb the interactions, which is represented on this left side of the plot. The surprise here was that we also had a lot of gain of binding mutations. Yeah? This we did not expect. And I will come back to what we think about them a bit later. But clearly, these mutations do create novel binding sites for motif binding domains, which surprised at least me. And the other thing we can notice when we look at this kind of plot here is that we have on the left arm where we have the motif breaking mutations, we have about 29% of the mutations affected previously known interactions, while on the right side, as expected, you can see that we have many fewer previously reported interactions. Now, this is still just a phage-based scoring system, so what we did was actually, we as in Johanna, she went and measured a lot of affinities, both of the wild type and the disease-associated mutant peptides, and we could see that while there was not a perfect correlation, it was good enough. So we can clearly link the effect of the mutations to the effect on binding by phage display. Now, if we look at this left arm here, we can also look at what kind of proteins do we find here. So what are the most common loss of function mutations? What do they affect in terms of binding partners? And many of these proteins were found, interactions were related to loss of binding to E3 ligases, such as KEAP1 and NED4. We also saw loss of binding to proteins involved in cellular trafficking. Just showing you one example here, we have, again, KPNA4, which I said bind to nuclear localization signals. For this bait protein, we found 3 different mutations here, which target the exact binding motif. So we have an R to C mutation here, a K to E mutation here, and an R to Q mutation here. And these mutations are associated with different diseases, 1 which is cancer and 2 that are developmental disorders. So just to go deeper into one of them, we have this mutation of CDC45, which is required for initiation of the chromosomal DNA replication. And we know that this R157C mutation is coming from this patient here. So these patients are getting specific phenotypes, if you want. by testing the affinity, what happens when we do this, introduce this R2Q mutation, we can see a significant loss of binding. So I think this was a 1,000-fold loss change of affinity. And we can also see that we do lose the interaction between the full-length proteins, and maybe more importantly, we do lose the loss of nuclear localization of CDC45. So this is just one way to show how we can use these mutational data, property data, to start to pinpoint what is actually happening, what do these mutations do. Now, coming to the other arm, the neo-interactions, this is was the more surprising results of this study, maybe. If we look at it, we can see that we have many serines, for example, being mutated to more hydrophobic bulky residues. And what they do then is that by introducing more hydrophobic residues or specific residues, we can get novel binding sites. So here we see a pathogenic mutation of BAB1, which is a mitotic serine threonine kinase, which increases its affinity for LC3. We also had a mutation of beta-catenin here, which turns an S to a phenylalanine, which creates an FG motif which binds to G3BP1, which is a core stress granule protein. We could nicely show here that we go from no binding to quite a good affinity here of these interactions. And we were a bit, we did try, I should say, to see if this mutation targeted beta-catenin to stress granules, but we were not very successful. So we looked a bit more into this, and we realized that this S33F mutation is also destroying the beta-catenin phosphodegron, meaning that what we see might be kind of an additional function of this mutation. Now, yeah, someone will tell me when I need to stop. I'm losing track of time, sorry. What is beautiful with this data set is that you can start to look not only at one disease, but actually at many diseases because of the way that we designed the library. So we have a lot of mutations, for example, that are associated to cancer, and then we can extract these mutations and start mapping them onto the protein-protein interaction networks. So you can play with it if you feel like it's published. What is also neat here is that we can start to understand a bit about motif evolution. So we can see that, as expected, many of the mutations that create novel binding sites are really creating the expected consensus motifs. And many of the mutations that are conferring loss of binding are also occurring in the consensus motif residues. That makes sense. What we can also see when we look closer at the data is that quite many mutations are actually not hitting on the core motif residues, but in the flanking regions, which are contributing to binding, and also what we call the wildcard position, which are the residues between the core consensus motif residues. Yes, I think maybe the more interesting or more troubling result is that we also have binding generating mutations for which we don't know what they do. So we don't know the consensus, so we can actually not know, or we do know the motif consensus, but the peptides that we have do not match the core consensus motifs. So we do think that the motifs matters, but that there is much more to this field, and that the field kind of, well, the field that's also in me, we need to start to go not only from looking at the motifs to also look at alternative solutions for peptides to bind to the same binding pockets, which we will come back to in a second. Yeah, so this is just to say that we generated a fairly large scale data set on how motif breaking mutations can link to diseases, and yeah, I think this is quite exciting. Now, since this is a deep mutational scanning meeting, I added something that I actually rarely talked about, but which was just accepted for publication in Protein Science. So it links to this thing that not all peptides that we find that bind to a certain domain have a consensus motif. Yeah? So what we wanted to do then, because we were a little bit jealous of you guys all doing deep mutational scanning, was to develop a multiplexed peptide phage display-based approach for a quite efficient high-throughput profiling of motifs. So to do this, what we did was to collect again we generated a bit of a bait protein collection here for which we know what they are expected to bind. We then went to our favorite databases, the M database and our own, of course, motif collection, and we picked parental peptides. And then for each peptide, we tiled the same region using two overlapping peptides, so they overlapped by 14 amino acids, and then we performed the classical DMS scanning, or we designed the library, which was then used in high-throughput selections. And as we didn't know exactly what to expect, we sequenced every day to evaluate how many days of selection do we need to do. And to cut a long story short, after analyzing the data in all possible ways that we could possibly do, we found that it's actually enough to make maybe 2 rounds of selection. We could get by making 1 round of phage selections and correcting for input bias. But to have a more robust general protocol, we landed in doing 2 to 3 days of selections. So just to show you one case, we will look again at this G3BP1 case, which is a very curious case. It's this protein that binds to FG repeats, which are found in many different proteins, and many of them are important stress granule components. It is also being bound by SARS-CoV-2, the N protein, as well as the NSP3 protein. And most of these ligands are having this FG motif. But there is also one peptide here called caprin, which does not have an FG motif, right? So we thought this was an interesting case to include because how does this protein really bind to 2 different peptides of different motifs? So just to start from the beginning, we have this USP10 peptide, which is here, where we have a classical FG motif in the middle, and through our DMS analysis, we do correctly get back the expected consensus motif. We have again, here's a typo, sorry for that. We have the N peptide from the N protein of SARS-CoV-2, and again, we can get back the expected FG motif, while the flanking region starts to look a little bit different. The quality, I should say that the quality of this DMS analysis, it gets better when the affinity of the parental peptide is better. Finally, we come to this caprin-1 peptide, and what we can see here is that it does not contain an FG motif anymore, but it does contain what is up here, an FI motif. So it doesn't like, within this context of the protein, the G is no longer the favorable residue. How does this work? Well, one can go to the structures because the community has been pretty good, and these are and solved the structures actually for all these 3 peptides. And what is very neat here is that what seems to be needed is the docking of phenylalanine here, and then there are different solutions on how to fit the flanking regions. Yes. Okay, a very quick thing. So I showed you the purpose of this first story was really to develop a pipeline where we could first do the property experiments, and then use that, well, for the cases where we found peptides that did not match the consensus motifs, or when we found only 1 or 2 peptides that were binding, then we needed something more to add on our pipeline. And this is where Emiliani comes in. Emily, she just defended her PhD and has been focusing on DABs, but here we will talk about her USP8 story. So USP8 is a very curious enzyme. It's a ubiquitin-specific protease with an important function in regulating protein trafficking and stability. Now, deregulation of this DAB is linked to several diseases, and well, it looks like this. So you have 2 domains here at the N-terminus, an MIT domain and a rhodanase domain, and then we have the catalytic domain down here. Now the WW-like domain serves some kind of auto-inhibitory function here. But our question here was really about the rhodanase domain. What is it up to? It had been described to bind to to proteins, but it was not really clear what it was doing. So Emily simply produced this protein, the rhodanese domain, which I forgot to say is normally catalytic, but this one is lacking a catalytic cysteine. And the question she asked was, does it bind to peptides? And if so, what is the consensus binding motif? So she conducted her experiments, and she could find 5 peptides, so very few peptides actually, binding to the rhodanese domain. Few, you may say, but there were no peptides known before our study that were known to bind to this domain. So it is a starting point. What we could do was to measure the affinities, and we could validate binding. For one of the peptides, we could also model the interaction, so we got this kind of binding site here. But for the other peptides, we could actually not dock the interaction, so AlphaFold kind of gave up and returned only very low confidence interactions. So then, having our new favorite tool, the DMS by phage display, we decided to characterize these interactions a little bit more. So we picked 2 of these peptides, so the rhodanese, the Kia type peptide and the TET3 type peptide, and we've performed the DMS analysis. And I think what is very neat here is that the analysis actually converged on 2 different motifs. So we now go from having one domain which was not known to bind to peptides, and now instead we know that it binds to different types of peptides, which likely bind to the same binding site because there is a direct competition between them. And for one of them, we can model the interaction, and the other one not. Anyway, we thought this was very curious, so we were coming back to the original question is of how does USP8 bind to its substrates? Is it so that this motif might actually direct the enzyme to its substrate? So to answer that question, we did a bit of motif scanning where we picked, we used the type 1 motif to scan USP8 substrates for matching motifs, and we could pull out that a couple of them had the full matching sequence, while many of them had partial motifs. And then we validated these interactions using peptide spot alanine scanning or peptide spot analysis. And then eventually we also validated that the motifs that we use for predicting the interactions were also important for binding. So this is our current very schematic picture of how the motif binding of the rhodanese domain might contribute to substrate binding. Now, for people of your field, of course, this might be relevant when you start to look at what the effect of disease-associated mutations. So the take-home here is that we have, over the years, developed a very robust, high-throughput platform for high-throughput screening of motif-based interactions. We have also developed a protocol for discovering both motif-breaking and motif-making mutations. And we have added on the DMS by phage display for the deep mutational scanning so that we can gain more information. For those of you that are interested in the interaction data that we will release soon, it will hopefully come in August. But if you have your favorite protein that you're interested in, you're always free to contact me. Coming back to my lab, here they are, still happy, still working, still on the roof, on top of things. We have funding, of course, and I don't know if this is appropriate or not, but I will take this opportunity to say that I'm looking for a new colleague. So we have an opening for a senior associate professor at Uppsala University, and if you want to know more, you can also contact me. Thank you.
Okay, we have time for 2 questions.
So for the